If y'all don't mind unmuting the room when you start the conversation, that'd be great.
Here. I just asked a question about how funding is might look under this umbrella moving forward and how engagement. If someone were to submit something in the next 6 months, what would that even look like? Is there going to be special opportunities? That kind of question.
So I think I'm not getting into trouble.
Yeah.
I can say that we would encourage investigator initiated applications within topics area. A notice of scientific interest,
Mhm.
because there's no commitment of funds. So you have to make a payment, either at NIBID or at MIA or at some institute. The process for creating a set aside, I can't say that we would create one. That's a limit on what I can say.
Yeah.
So that's that would the earliest that kind of thing would come out would be 2 weeks.
Yeah. Okay. So that's away from here. Yeah.
So we would try to encourage investigator initiated applications at this time. Then the institutes have either select pay discretionary funds. There might be possible hold things in that are beyond the institutes. That's the practice.
Yeah. So I talked about seems unlikely or less likely, not unlikely is too strong a word, that proposals of this type going through CMAD, for instance, would make the pay line, just because people be like, what are you doing? What is this about? Or it's not mechanism specific or whatever. So can we create a strategy or language that would get it to actually go to the systems synthetic biology panel, because apparently one does exist that would be more sympathetic to interesting applications of synthetic biology than an aging panel that would know what synthetic biology was.
So that's a separate step process.
Yeah.
One way of going about it is people who are interested in these topics contact the SRO for that study section, volunteer review, and what it is that they want. It's just ad hoc.
Yeah.
How the discussions go, what works and what doesn't work, and then when you create an application, contact the scientific review officer with your specific aims. This seems to fit with the review panels and as program officers, we have a different take on it. We think about how reviews go, and whether no one knows what aging is, and pass through grant on aging. You know, you could structure it there.
Yeah, well, that's it. All new. I'm through the cycle in many different fields. All new emerging fields fall between the cracks, right? And so what's the strategy for sometimes you go into a panel and people are like, this is the newest, greatest thing ever, like, and then you get a great score because people don't know what you're doing. But that's happened to me a few times. But it's
so seeding a field is always difficult to just I can tell you that the holding this meeting is organized by both NIBID and NIA indicates a very strong interest.
Yeah.
But what we can do with that is, for all things, there are competitions. So we might think this is the greatest thing ever. And our institute directors think, well, yeah, it's great, but that's the whole great.
Exactly.
One painful example for me was systems biology, which I promoted a long time ago. And my division got $800,000 worth of grant money. So, how are you going to do anything with that? And then,
But we're very grateful for our little fraction of it, yeah. Several of us, I guess, yeah. Yeah.
but then the tens of millions of dollars have been poured into it in Alzheimer's disease. So I feel like I'm still standing there by the door, holding it open. Everybody else that's walking through it.
Yeah, I think
David, we were just talking about this conversation that we had about the strategy, how to align grants so that they actually get reviewed and have some chance as the, you know, it's the launch part of this kind of integrated work that you have to find your way through when there's not obvious nurturing ways of doing it. And Ron just said it would be several years. If such a thing were to emerge, it would still take several years for that.
As a set aside of funds specifically to see the field. On the other hand, here's something I don't think anyone's tried. It's probably a bit subversive. That is, so you guys decide, you know, these look like they're interesting things. You keep in touch with each other and you come in as a flotilla. So the positive side of that is, I mean, they're not grants that are specific linked to each other. It's just you have a whole bunch, an armada coming into port. And so they get up in the study section and that draws attention. Here's an area that's emerging. Look at all the grants that are here. So that can be helpful. But the downside is you're competing with each other.
Yeah.
So how altruistic do you?
Like in the Armada, the artillery turns towards it and it takes it out.
Right, the currents coming firing on each other.
Yeah.
Did you mention the cellular molecular technologies study section?
I didn't mention one specific.
I mean, that one has been a locus for synthetic biology past several years now. You know, yeah, I don't know the aging biology panel.
Well, there's one.
Yeah, so
Yeah, but
it may be a bit orthogonal to that. Obviously, you know, I can't
so,
speak to
you
it.
know, there's another way of doing it, which of course is you don't use the word aging. You don't get people who don't know what aging is.
Each study section you mentioned.
CMT, cellular and molecular technologies.
Okay, so that has a synthetic biology.
It actually uses the word synthetic biology in its description.
Okay.
So if you wanted to do synthetic biology directly against one of the hallmarks of aging, it would be, well, this is synthetic biology. Gene misexpression. Autophagy. Something like that. And parenthetically, and it's one of the hallmarks.
The caveat is that you don't control what goes in the study section.
So
Or
David,
CSR stuff.
Patrick asking us, you know, future funding situation for this area, maybe you can say something. Exactly about an idea.
This is, you know, a topic that's of interest to both NIA and NWIB. So the NOCI's out there right now. So use the NOCI, right?
But so could you just expand on these notes? What do they mean? Like did they give you what?
It's like ringing a dinner bell, right? It's like a vector, right? It points to the parent R01 or
Yeah.
the parent R21 or the parent R03 or whatever that institute.
At what point in the process does it weigh in on anything?
Programmatic. It
Yeah.
may,
Yeah,
right?
it just says that the program is interested in these kinds of things.
Yeah. Now, CSR does not.
Through the
do
review
it.
process, there's no fight or anything like that.
So in principle, the NOCI would the encouragement to a specific group, administrative group. To use. To reach beyond the percentile pay line doesn't make
I
percent.
see, that's
So
because that panel doesn't know, like, because I've
So the
been on
panels-
panels where, sorry. Just the notes, where you have special programs instructed as a panel member to whatever.
that's right, that's
That would
right.
never happen with the notes.
There's no special review
It's not
criteria.
high, it's not highlighted during the review process that this NOC exists.
I mean, somebody might mention it, but it's irrelevant because there is no special review criteria
Right.
attached to the NOC. Now, NIGMS, for instance, they use the NOC, they point to their Mira applications. I believe the Mira has some special review criteria because I believe it goes to a special emphasis panel.
Yeah.
So then, of course, that special emphasis panel will review it with its special review criteria, but that is particular to that Mira. Whereas like we point to the parent RO1, there's no special
That's
review
that.
criteria attached to it.
Sorry to wonk out on
No.
this, so
No, I mean,
What
I
did
thought
you
there
wanna
was,
say?
I thought there was emphasis in the panel on nosy applications. And maybe I was just totally missing. Like if, like a nosy proposal goes to CMT, then there'll be, okay, now we're looking at nosy app. No,
I don't
Okay,
think, I
I
don't
was
think review
misinformed.
officers are beholden to point out nosies, but there are so many nosies. Out in the space, you know.
Yeah. okay, okay.
Yep, there's no special review criteria unless it's dictated
A lot
by.
of urban legends
by
out there
what
about
it pointed
nosies.
to.
well, that's why, I mean, it's an interesting process. It feels to the investigator like it should be a bigger deal than it is, so, for sure.
So in general, there are three kind of funding opportunities, right? One is RFA, which has a set aside, and also special emphasis panel. So that's the best, right? So the next one is for the PAR. There is a, a special review, right? So, but there is no set aside.
Right,
But you will have, expertise, in the special emphasis panel to review, this topic.
It doesn't
Then-
even have to be a special emphasis panel,
Right.
but the review panel should pay attention to
To the-
the review criteria. in it.
criteria of the.
So it could be, part of existing, and,
Yes,
but, with a kind of set aside time,
so I've done that before.
right?
So,
Then the third kind, program announcement and those with some kind of, emphasis without money, without special emphasis panel.
Okay,
One of the advantages, so if there is an OC. And a lot of grants, I don't know what's a lot, 6 come in on that OC. They can be directed to one review panel. Then program officers can draw attention to that. Yes, say, you got 6 of these, have you got the expertise? We could. Recommendation,
yeah, that's the way to do it.
but then you build it, so it's a building process.
And you, the PI, can also make that inquiry to the SRO too.
Yeah.
Hey, I ended up in this panel that I really didn't, it has nothing to do with synthetic biology and it's nothing to do with aging. Why am I here? Right? I mean, you ask those questions. That's where you get shoveled.
But super helpful, thank you. Sorry to, but at some point, if we're
I
gonna
just
do
popped
work,
in
we
to see
need
what
to get
the
money to do it.
breakdown was, like, this room's got like 7 of you, and I was gonna like, and the other room has like 3, so I was just curious as to what the breakup was. On to see how it is.
So I think the basic tools proposal to CMT could work, but and then, aim 3 would be aging something.
Yeah,
But
that's
then.
what I think too.
Yeah, but I mean, I would be interested in exploring the aging. As a possibility, like maybe we could talk to the SRO. And see if there's, how far off base are we?
I think our best bet would, well, I don't know, have you served on this cellular and molecular technologies panel before?
I'm set to, but they haven't called me up yet.
Okay,
I'm funded, I was funded by CMT.
Yeah.
Yeah, twice actually. So I've got a pretty good feel for what they look for. It's a lot of protein engineers on there. So you think about doing, basic circuit design for, some health application that shows up by aim 3. That those tend to be favorably reviewed.
I mean, I would think if people are paying attention, developing a novel synthetic biology system in the whole animal is a new thing that could really help move the field forward.
Yeah.
And so just on first principles.
Yeah, and then the proof of concepts could point towards the control of gene expression, tight control of gene expression across the organism for aging models. It doesn't necessarily have to propose the work. But you know, again, that could come in three. It's like we're gonna try to express or not express, try to do like some of the basic circuits like that we were talking about last night.
Yeah.
By AIM-3.
That's, I think that's right. I think one of the questions, because again, it's. Would there be separate money or opportunities or whatever is there, is there a is there a model or circuit analysis as well as design? Have you done that in your work in yeast? Has that been a quantitative
separate
analysis to say, oh, this is what the circuit should do, here's what it does do? Measure flux through the circuit or something like that. Have you done that?
Yeah, but that's regular R1.
Yeah, no,
Yeah.
that's what I'm saying, but.
Yeah, definitely. That's all I did. So, yeah, definitely. I think another, yeah. I never tried CMT. Is that a good panel?
Yeah, that's been the gateway for synthetic biology,
Okay,
like
okay.
beyond NIGMS. That's
Okay.
been it was a lot of proposals where Dave basically helped people write Ames pages so that they would go to go to that panel and get reviewed, and then synthetic biology people started showing up on it, you know, kind of
That's
after
nice.
the original panel, which was mostly protein engineers, had already approved like a
Uh-huh.
few R01s through that. But,
Okay.
yeah, that was I crafted my R01 so that
Okay.
we could get it straight into that panel, and then, you know, there's people that will review it and, you know, 100% understand what the goals are. So,
Great.
but it's very much about tool development,
Okay,
right? And,
Okay.
you know, it's kind of consistent with the mission of the NIBIB, which is, you know, to engineer tools and modalities for doing therapy,
I see,
right?
I see,
So,
I see.
I mean, the aging models is the motivation, you know, there's a possibility, but I think as long as, you know, you keep it in the, you know, kind of the tools building wheelhouse, that's probably reasonable. I don't know, be curious to see what Dave thinks about that idea, but
Yeah.
I mean, I think the crafting you know, SynBio proposal for an aging panel is risky without the circuit already in hand or without the molecular
It's not
that's already in hand.
The transform development
Mhm.
is to synthetic biology, the whole animal
Yeah.
in a comprehensive way, especially if we can use some combination of the combinatorial power of doing lots of animals,
Mhm.
whole genome approaches, and then being able to integrate whole library kinds of things. That seems quite innovative to me.
I mean, I think that the argument he made that, you know, we can go through all of the evolution of parts discovery and, you know, understanding composition really quickly using these platforms, you know, in a way that E. coli and yeast offer obvious advantages. You know, you can pick colonies, you can put colonies in wells, right? You know, never really had that capability with C. elegans until your guys' platform was developed, right? Yep.
Yeah, and that's been our central argument, our theme that we're trying to build
Mhm.
is basically making C. elegans a microbe. And the challenge for doing that was microbes, you can take the DNA from the outside and then put it through the membrane. In an animal, you can't do that because you have the germ separation
Yeah.
from soma. So you have to put the DNA in first and then use the library. But the logic, the approach, everything has already been developed in the synthetic biology of bacteria and yeast. That would be the argument.
I think that's-
Translate that.
That's really strong.
Yeah, it's huge component of it is exactly that, that we can just take everything, the entire synthetic biology world building, but now we can put it into the work system through that, because there's a massive amount of tool software, things like that. There's already been, you know, all this. That, but we now kind of, okay, well, let's just now put that straight into this animal model. And I think, yeah, I mean, one of the things that we want to even think about, particularly aging model, like you were mentioning, the thing, we could create like a model of aging, maybe even say in a tissue, like a mitostress kind of thing, right? Like, I think that could be proposed. I don't know, kind of panels or things like that, because I'm not as familiar, but if it was like, you know, a mitostress thing, we could potentially make, you know, a transgenic that has like a mitochondrial tag, causes stress to the mitochondria, and that's only in the neuron. Or something like that, and then do, you know, drug development, patients like that to help
Yeah.
alleviate that and do that in a fashion.
That wouldn't be the first proposal, but
Sure.
that, I mean, those are things that can be done. I think the first proposal would be to kind of build the tools and framework for.
Yeah.
Doing circuits and then. Combinatorially, how big a circuit can you do, and then what's the most interesting circuit? And then can you have these artificial promoter systems so that you can. To me, the outside perspective on the advantage of the synthetic biology is you can bring things from the outside that you can manipulate in a way that won't generate this crosstalk issues with internal systems, and so have things be as fully synthetic as possible.
Yeah.
And then also synthetically have specificity targets. I think it's the power. It
We
feels
call
like-
that, we call that broadly orthogonality.
Yeah.
Broadly or not, that's-
Yeah. So I think orthogonality.
It means several different things, but that's exactly what you're articulating.
Yeah, so I think that in the worm world, there's been no orthogonality.
That's an
People are
interesting
fully
statement.
within the
Yeah.
biology of the system and articulated,
most-
all these things are tropic.
Yeah, I think most, most traditional geneticists. It takes them a while to catch on to the advantages of building an orthogonal system as a perturbation. I think that may be a little bit not surprising.
Well, it sounds like the best part is just. It's just, you're building the system that's completely almost encapsulated with the rest of the environment that you can now control.
Yeah.
Want to respond to it.
And I mean, I think the thing you get hung up on is, you know, of course, it's never true. It's never fully insulated.
Sure.
It's functionally insulated to the extent that you want to perturb the system, right? And I think that's the thing about, like, you're putting this other gene in and these other genes here, and, you know, now you're messing up the cellular function, right?
Sure.
And, you know, cells can tolerate transgenes, right?
Yeah.
You can put them in and you can do perturbations with them. It's fine.
So what are the most interesting circuits? Or what, so what's the parts list of things that exist?
Yeah.
For your approach is like, let's discover parts. Right? So,
Yeah.
But there's still the initial components, there's the baseline that you've got to start.
So, I mean, you need, you know, you need the basic parts, you need things to encode the genes, right? You need promoters and you need terminators. And then you need things that will, you know, make circuit connections, things that'll give you the dynamics. So you need the transcription factors that interact with the promoters. And, you know, once you have those, then, you know, you have a toolkit of those and you can start building stuff, right? So you get into activation versus repression. I mean, repression is something that's leveraged in bacteria, certainly. You know, animal promoters work differently. It's mostly about activation, right?
Yeah.
So we mostly think about ways to turn things on. So then the trick with control of expression is to keep things that you want off, and then to turn them on inducibly. When you talk about the transcription factors inducing, then there's small molecule control, optogenetic control, and then, you know, other means of control that maybe tap into or are connected to endogenous processes or endogenous networks. So I'd say those would be the three categories of control for gene expression that I would say would be useful to develop technologies for.
So we've been talking about it a little bit, you know, before the meeting, and that I don't know if there's maybe a more naive perspective on it, but since we don't do the development of like transcription factors and things like that, just, you know, basically tooling the CRISPR-A, CRISPR-I system to become these sort of artificial.
We could do
transcription
that.
factors.
That's
Is
one
that sort of
strategy,
push,
yeah.
Or is that?
No, I think that's one strategy. Like CRISPR didn't evolve to be a transcription factor. It's a hack, right? And it's not, you know, as a hack, it's a good one. You know, CRISPR takes a little while to jump onto a piece of DNA. It takes a little while to fall off too. So, you know, it didn't evolve like, you know, RNA and protein as a combination didn't evolve as something that's a transcriptional regulator. right? And it's these small protein domains that, you know, bind to relatively short sequences that evolve. So that's my take on like where do you start with the transcription factor? You kind of start with the design that's already there. And so from that standpoint, you know, I would say zinc fingers are very programmable, very easy to work with. They always bind tightly. And I think as transcriptional activators, there's more examples of transcriptional repressors in nature with zinc fingers, but there's plenty of examples with activators too. So that's my toolkit of choice for anything outside of bacteria, basically. So, you know, working with zinc fingers, then you can imagine like figuring out what is the best activation domain to use and a combination of activation domain and core promoter to use in C. elegans. And these are things that we can interrogate with a library approach very easily. And this would be, you know, a little bit pedestrian in terms of this, and you know, it's like we're just trying to find the basic parts here. But then once we have the basic parts, then we can, you know, think about circuits, think about inducible control or optogenetic control, or connecting, again, making connections between endogenous processes and expression. So I think it's one way to motivate it is like, you know, what would we want to build that would be, you know, either external control of aging related genes, or would be, you know, some sort of autonomous control where we're sensing and responding. We're creating that control loop that I think somebody was talking about earlier, that's changing aging phenotype, you know, maybe in a tissue specific fashion. And then walk backwards from there and ask like, okay, to make a circuit like that, now what parts do we need? And I think the ones I just mentioned are probably sufficient, right?
Yeah.
I don't think we need to go too much further than that. I don't know. You built circuits. Does that track?
Yeah, that sounds perfect. Sounds great. Sounds great.
So we have this transcription factor DAF16, which is fork head box protein, which I don't know what that means in terms of how it actually binds DNA or what it does. But those motifs are known. I don't know if you could create an artificial zinc finger that could target the same
Spot.
motif or not.
So typically what we do is, if we have a double-stranded piece of DNA here, we'll put a minimal promoter, which is all the binding elements minus the activation domain or minus the enhancer domain, things like that. And then we'll put known DNA sequences upstream of it that our engineered transcription factors bind to. And then on the transcription factor, we fuse the activation domain of whatever functional transcription factor works. So we use, now it says it's BP64. 65.
Isn't it just that?
Usually, yeah. And then this has all the elements that it needs. You put your reporter or whatever your transgene is, your terminator.
I guess I'm thinking, so that's the, how you built the circuit in the first place, and I think that thing one is like, you have that is the GFP, whatever, and when you're building your artificial circuit.
The part that
The aim
we-
3 that I'm thinking of is instead of GFP,
Yeah.
can that be a transcription factor that then targets these the stress related DAF16 things,
Mhm.
because then you could have a circuit that's activated in specific tissues, or certain controls. And then ideally, what you would like to do is where I think that's what I want is, let's say you got a set of 300 genes, you know, they're all have the DAF16 binding sites. Is there any way to only bind one of those genes at a time?
Mhm.
Based on whatever you're attaching to the end of that circuit, so it kind of system through, so that you could activate things one at a time systematically, and then get at the question, which of these 300 genes is the most important, or what is the fraction that is contributing to longevity extension?
Yeah, what subset?
Yeah, so
That's
in
great.
which tissues? So people have done tissue specific DAF16, and it looks like the intestine is the most important tissue for things to be expressive. Well, what genes in the intestine? Why the intestine?
So
Why
that's,
is the intestine drive longevity and the rest of the worm, those kinds of things.
that's, I think that's what we were looking for, but that's the motivation for the toolkit.
Yeah.
That's great. Okay,
I think we totally do the single one at a time, that's the adaptive target drive, we could just do the split integration, single copy approach in a tissue specific fashion, right? So like we had a 300 guides or transcription factor things, but they're all targeting an individual one. We just do the single copy.
The question
integration
is,
of those.
can you do the targeting? Yes.
Yeah.
I'm confident that we can make the libraries and do all that sort of stuff, but it's like, can we actually? And if we can't, then we can, there's lots of different ways of creating more specificity. To me, that would be the holy grail for. For right now, that would be transformational if we were able to do that, because then we would both create artificial circuits. And then we would use that artificial circuit to probe in a precise way, things that have been known, but are the rest of the aging related network.
Okay.
So we start pulling apart that network in a precise, using precise tweezers. That's one way of thinking.
is there, I mean, is there a need for, or any value to temporal control over
Yeah.
expression, overexpression of these DAF genes? Like at some, like different stages in the worm's life cycle?
Exactly, or different, I mean, I think one of the really important questions that's emerged from our work on drug development is how late can you do something and have it extend life? So if you turn on these DAF16 thread, let's say we are able to identify, these are the
That's
subset of
great.
genes. If we, turn them on in day 1 adult, for sure, it's gonna make them live. What if we turn them on in day 12 adult when these things are like already pretty creaky? So that's basically what the gets at kind of what the human aging paradigm is right now. All of our money and all our interventions are in old people. And I think there's a fundamental biological question. It's like, can that really make a difference? Or do you have to start with 30-year-olds in order to have these long-term health impacts? Because if that's the biological reality, that's going to totally change the way that we think about it. Then if we can, it's great if you do 30-year-olds, they'll live a long time, but it's okay if you're doing 60-year-olds, 70-year-olds, 80-year-olds, you'll still have these massive benefits.
I see.
I'm actually dubious that that will be true, but it's a testable hypothesis.
Oh, that's great. So how many different tissue or cell types, totally ignorant of
Yeah.
biology, are there, and would we want to have specific control?
And we do have, orders that are specific. So there's big
Oh, okay.
classes of things, intestine, skin, the neurons are kind of the major
Yeah.
groups. There's
Germline.
germline, there's excretory pores and things
Yeah.
like that. You can get pretty micro
Yeah.
into it, but there's not a gazillion, there's only 1,000 cells. So there's a finite number of them cell types within that class.
How good, I mean, how specific are they?
They're pretty specific, but they're also temporally, not always the same expression across the whole thing, right? So one of the synthetic things that'd be great is to be able
That
to
would
be
be-
tissue specific, but constant or
Yeah.
more programmable in terms of its intensity.
That
So-
wouldn't be, I mean, that wouldn't be at the top of my list of cool circuits, but something that but it'd still be a challenge, but something that normalizes expression across the tissues
Yeah.
then.
Yeah.
Takes a noisy input and, it's probably uses some combination of feedback to tune to the homogeneous expression level.
Yeah.
I mean, that's something that, again, could be generated with a library approach, right? And I think this is something that we're kind of exploring in the lab now, is it's hard to make tissue specific promoters in human cells. And it's mostly because it's hard to make promoters because we don't really understand that well why things turn on a lot or very little. It's mostly because expression is controlled over long length scales. And three-dimensional, there's three-dimensional effects too. So, with CLs, maybe there's more known, maybe the kind of the determinants for high expression and low expression are more compact over a shorter region. Who knows? But, one of the things that we're trying in human cells right now is installing positive feedback, with the zinc finger. So you have just a slight trickle of transcription coming through a tissue specific promoter. So you get specificity and you might have a decent handle on that, but you're not able to really tune the amount of expression that comes out of that promoter. So the little trickle can then be amplified with the positive feedback loop. And this is, you know, this is easy to engineer. This is still a compact circuit. And that positive feedback gets you up to, you know, basically maxes out your output promoter. And so then, you know, any little trickle of input from the tissue specific promoter gives you a normalized and really robust output expression. So that's a possibility for some, and that's a tool we could use too, to just say blast expression across all of one cell type.
And I think it comes down to some difference in development of mammalian versus worm cell. So
Yeah.
because there's determinant to develop, there's a commitment to a cell fate. Need expression that's cell specific, but then when that goes, you don't need to maintain that fate so aggressively with expression. Whereas in deuterostomes,
Yeah.
you got to keep the cells can keep changing their identity apparently, right? And they have to talk to each other. Am I still in the skin? Where am I?
Mhm.
So you know, whereas the protostomes are like, I am a skin cell. That's what I'm gonna do.
Yeah.
And, I don't really need my friends to tell me what I'm supposed to be doing.
Do worms have DNA methylation?
They don't have DNA methylation. They have a histone
They
modification.
just have histone modifications. Okay. It's good. That's,
Is that good?
well, it's, I mean, it's
That's-
easier to work with transgenes when the DNA doesn't get methylated.
Yeah.
Well,
Yeah,
they do
there
have
is silencing.
Yeah.
So piRNA is a big problem.
What's that?
The piRNAs, siRNA.
So we have the RNA based silencing, so that.
Oh, right.
So that's
Okay, I forgot
that
that.
was, discovered in worms, although. plants and
Okay.
people So had that.
Yeah.
But that's our big challenge
Yeah.
for expression.
I
is that
understand. So you got to stick in
Foreign
and
things
everything.
are,
Exactly.
Yeah.
yeah, yeah.
Oh, that's interesting.
It has to look natural.
Yeah.
Yeah, and there's been a huge development in that area over the last 5 years in particular.
Yeah.
People are figuring out the rules to basically fool this
Yeah.
internal immune system.
And but most of that's also just restricted to the germline. And so we can do, you know, native tissue somatic expression, usually a lot simpler. It
Yeah.
doesn't have that same silencing. problem.
Okay.
So a lot of things have been interested in the germline. And so unfortunately, a lot of the transgene silencing happens very robust in there. But then, yeah, tissue.
So that's an issue for us because we want to do germline editing.
Yeah.
In an activated state, but for the phenotypic downstream
Yeah.
effects of a circuit, it's not a big.
Yeah, exactly.
I understood you correctly. Maybe I'm just trying to draw this out. I understood there was two parts that wouldn't be great if you'd like to be able to do. You'd like to be able to have user-controlled expression of X gene. And then simultaneously do like a genome-wide knockout or a targeted knockout or knockdown of specific genes that are in various pathways on an individual gene by gene basis.
Yeah, actually, what I'd like to do is upregulation.
Upregulation, sure.
We can do knockdowns ourselves, but just genetically.
Yeah.
I'm confident that we will, in the next 6 months, have a strategy for that. But specific activation or overexpression is, I think, a challenge. And because of aging specifically, knockdowns are great for, as I said, making inferences about how to screw up development. And once you do that, then you can figure out what the positive thing is. But how to make things live longer presumably involves activation of genes at some point that don't want to be activated under the circumstances. And you'd like to do it in a precise way and the smallest subset that you could get, and then so on and so forth. So that there's no systematic expression. So the overexpression things have been done in work so far. Frequently it's just done by copy number.
Yeah.
So you can just inject a buttload of the gene
Yeah.
in as an external array, and then you assumed it's overexpressed because you got a lot of copies of it.
Yeah.
I see.
And
It's not
very
precise.
stochastic,
Yeah.
Very imprecise mosaic because of the lack of correct inheritance, but that's traditionally how overexpression has been done, and it's very, very easy.
Okay.
The other ways of done it, we do have very recently like TED-on, you know, expressions like that, but that's very leaky. So it's not very extremely reliable. We do have
Yeah.
like the Galford QAS systems that do work, but
Mhm.
again, how do you know, it's built on the drive for the Gal4 has to have, you know, expression there.
I
So
see.
there's not really a really great tunable overexpression system. It's kind of just like big or none, and then mosaic. And so happened to be ones like, oh, well, I want 2x or 3x or something like that. That's a lot harder to sort of get. Now we can just do 2 or 3 copy number, but then context matters too, where we put the 3 different transgenes impacted, things like that. So
Does CRISPR A works in, it works fine, right?
CRISPR activation does work. Yeah, we do. That's like just the last couple of years. People.
Okay, so having like a CRISPR A output array would be a way to do this probably.
That was what I was thinking.
Yeah.
Is having maybe the zinc finger system running the tunable
Mhm.
expression and then either have them linked or have in a separate system.
Yeah,
CRISPR A to do the targeted activation.
yeah,
Because it would be very difficult to do the protein engineering required to have a library of engineers that targeted all genes.
you know, that's a non-starter. That would be, you know, we'd have to start a separate research program for that.
The combinations are really cool paradigm.
Yeah, I guess the question of can you do guide arrays and how do you do guide arrays in C elegans? I mean, it's probably something that,
Well, we're working on it now.
okay, good.
I mean, so that's kind of the thing, right, with the TARDIS-based system here, we're inserting the barcodes, doing a bunch of those, in parallel, all single at a time, right? A barcode's only 15 base pairs, but a guide is only 1.
Yeah,
And so doing it fast.
oh yeah, the array could be the barcode. Wonderful.
I mean, the way that I would see it from a progression of action, then you can imagine the scenes, although this is dangerous because the names build on each other. So be careful. But you would do the initial circuit stuff, GFP or whatever. Same exact stuff that you guys have already done, using the same approach,
Yeah,
the same library stuff. We need to understand that better in terms of the combinatorics and and then whatever, how that progresses, maybe into something that's a little bit different. And then aim 3 is you just replace GFP with whatever this CRISPR A targeting,
yeah,
however that works, and then you're just going to, instead of upregulating GFP, you're gonna regulate a targeting system, that then's gonna turn on a gene of interest. And as long as you're targeting this known subset of unedited genes, then you could do that. There could be that and issue specific manner. That could be a sub-aim, it could be a separate thing, whatever. But to me, that's kind of a no-brainer.
yeah. I mean, the thing that's missing is I think that's good. I mean, just trying to think about the deliverables for each aim. The deliverable for the first one would be just, basic parts for expression control, and then second one would be the array capability. And so then, if one of those doesn't work, then what do you do, right? Well,
Yeah, so
aim
the
3 could be, well,
so could aim 1 just be, we know that here's the circuit.
mhm,
We don't need to evolve the circuit. We don't need to, like, here are components, work.
yeah,
We're just gonna engineer those and then have this tunable thing that can be activated with known components and turns on GFP. And that's deliverable. Even if we delivered that to the community, to the worm community,
Yeah.
you got a new way of activating with a small molecule,
That would
a
be-
gene.
yeah.
okay,
People would be like, okay, give me that strain.
Yeah.
And then we'll just swap out the GFP for whatever we want to turn on and
Yeah.
no, that's
and
gonna
that
help.
would be good. And then the aim 2 could be, all right, let's use the optimal circuit using your the approach that you guys have. And
Yeah.
if that fails, we still have the first thing, but we could maybe do something even better.
Yeah,
And
no, I
then
like
aim
that.
3 would be to use either the aim 1 system or ideally the aim 2 system to create this targeting array. So that,
I think
that's a way of
good.
doing it.
I think that's good. Yeah, that's good.
So I have 2 quick questions then. When I understood in the beginning, you wanted to butcher the name, the DAP. something.
Yes.
DAF 16.
Do you want to express DAF 16 and do the CRISPR-I, or you want to use CRISPR-IA to target one of the DAF 16?
CRISPR-I to target the DAF 16 targets.
Okay, so I was thinking we'd be expressing DAF 16 and then interrogating the rest of the genome separately.
Well, I'm not sure. I guess there's a separability issue. So the way that DAF 16 works is it's always on. It's just sitting in the cytoplasm. And then there's a cascade, series of kinases that under certain circumstances will phosphorylate and dephosphorylate DAV16 so that it'll migrate from the cytoplasm into the nucleus and then it turns on.
Interesting.
So it's always there. Now, if you want to have it not be there, actually. Actually.
Yeah.
And then, but either way, then what I'm saying is that screw DAV16, let's have our little thing in the nucleus. Taking it, basically a target at a time.
Okay.
I mean, more broadly, in the scalability, why stop at DAV16 targets? Why not just target every gene in the genome and then ask whether or not it extends life? You know, so on regulation, that's what we want to do. So,
The
but you
second
could
question-
start with DAV16 target.
The second question I had is you mentioned there's no steroid hormone signaling.
That's right. So there's no testosterone. It's true.
So the main function of our circuit that works really well is it's an estrogen analog that binds together two components and then brings it through this.
Yeah.
to the cell. And, sorry, from the, you know what I'm saying? Right, it's happening today.
Right.
Do you anticipate that that would give us any trouble, given that they don't have steroid signaling?
Is it,
No, I mean, it's effectively orthogonal, right?
And that would be even more.
maybe even more orthogonal, because
Yeah.
it's like,
No,
that thing doesn't exist.
Yeah, I mean, invertebrate steroid hormones are, yeah, that you can use them in, like lower organisms, it's basically orthogonal. Yeah,
So that'll be better.
Yeah.
yeah.
I just want to get in the way of anything else. It'd be isolated.
It's mildly toxic, right? 4-HT.
It's mildly toxic to human cells. I would imagine that I mean, yeah, if you put it in its micromolar concentrations, I'm sure it would be, but I would predict that the worms just swim around in it and don't care.
What's the drug?
4-hydroxy.
Tamoxifen, yeah, it's just,
Chemotherapy, right?
yeah, it's just tamoxifen.
Interesting.
Essentially are the two split proteins bind to it that relies on the estrogen receptor translocation that is native to.
Yeah. So that's it's a question of whether or not the chaperones that C. elegans have will keep it out of the nucleus or not when it's disordered. And then it reorders around binding to the hormone and then goes in via now an ordered NLS sequence. Okay.
But it's a diamond.
No, it's a monomer. It's a
It's a
monomer.
monomer,
Yeah, yeah. You know, this works in yeast just fine.
Oh,
Okay. that's
Yeah.
fine.
Yeah, because NLS and NES signals are pretty much universal. Excuse me. And I think it'll work. I mean, I haven't explicitly seen it used in worms. It's been used in flies though. You know, and we have a couple other inducible systems too that would work great.
That would sound, I mean, just that, yeah, that would.
Yeah. We can also use abscisic acid, which is, I think, another thing that the worms probably don't care about.
That's the kind of thing,
Yeah.
like, we could just have somebody in the worm community say, hey, if we had
Yeah.
this, attributable, tamoxifen-based gene expression, that would change
Yeah.
the way that we do our work.
Yeah.
Because I think just that would be
great.
a huge contribution, just by itself.
Yeah.
That's a synthetic biology.
That's
And you're
totally
just
the orthogonal.
remapping, whatever the single cell approach to a multicellular organism. It's kind of these things are kind of no brainers, but that's why we're like. All right, this is a moment in history where there's such an obvious thing to do.
Yeah.
How many other people are doing this? Because I think that there's literally only one other, but they're not thinking about
They're
this.
not thinking about this side.
Yeah.
They're not.
So no one is really that we can tell. Thinking about how to utilize this next generation of genome editing technologies to create synthetic biology. There was I was just at the worm meeting. We were both in Glasgow, Scotland. One of the reasons I really wanted to go is because there was a workshop on synthetic biology and worms. Thought, oh, we got to figure out what this is. It was just neurobiologists talking about optogenetic control of neurons as a synthetic biology, which to me has nothing to do with synthetic. I mean, it has something to do with synthetic biology, but
that type of stuff was around before synthetic biology.
exactly.
And, hasn't really evolved with that much.
Yeah.
So I would argue that it's none of the same people. I mean, you can call
No,
it,
Not at all. As a matter of fact, they opened the thing saying, many people would not call this synthetic biology. And then they went, I'm like, yeah, I'm in the audience. I don't want to be here right now. So like, you guys.
so I mean,
Good.
earlier when the whole group was in the room, I might have put my foot in my mouth on that, but I can't think of anybody doing like basic expression control gene circuits in C. elegans.
I didn't, so I was watching Kamala and Tara, they both were agreeing with you.
Yeah, I mean, I think I was just kind of realizing it's weird that that's true. And then, I think, yeah, I mean, I think it's just a huge missed opportunity on our part.
Like, it's just, because I've been looking at this now for maybe the past 6 months-ish or so.
Yeah, you would know best.
I'm just looking at this big open field, like there's a low hanging fruit tree just sitting there in the middle of it and all the apples are right there and everyone's walking around. It's just like, nobody touching it.
But that's, I mean, I've been in the C. elegans world for a while here. That's classic C. elegans
Yeah.
biology. It's like literally, someone says, here is a new hammer, and then people just go whack, 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 and then every once in a while, someone says, here's another new hammer. I mean, there are important things like RNAi and things like that. But there are other fields like in yeast biology or fly biology where people are hammer makers, like that's their job. The worm people
There's
are
no
not
hammer.
that way. Someone kind of, oh, I found this weird thing,
Yeah.
and then, or we adopt something like CRISPR. Worm people are like, oh, CRISPR,
Yeah.
we've been doing genetic transformation. They immediately applied it. But it's not like the development of CRISPR technology is occurring in worms. People are
Right.
just like, oh, now we can do this in worms. Now we can do this in worms, but they're not CRISPR biologists of worms that are using worms to develop new CRISPR technology. It's just not the way it happens.
I mean, there is the one place where there's a little bit of overlap is people using C. elegans as models for antibiotic resistance and microbial consortia, which is, I think a little, I don't know if that's died down, but that was kind of a thing like 5 years ago. You have a colleague who's engineered sense and response circuits in bacteria and then sent them into C. elegans gut to try to detect inflammation.
Yeah.
But that's it. That's really it.
Can I draw something really quick and just make sure I'm understanding, like what would be the awesome goal here for you guys? So you would like to have maybe something that's driving tissue specificity. That is then maybe this is putting in the zinc finger.
Your next goal, like if you have this tool, how do you,
That is
what
driving-
output
so
do you
this
have
sits
and
down
what,
here,
how do you
maybe the
analyze the
cosmic
network?
network.
Well, that's the
what-
question is
Yeah.
like, do we then start trying to
A, and then that gives
flesh
you
out the whole network, what
Yeah,
the regulatory
exactly.
things
big
are?
question.
I guess the
Then you
question
have to
is
use
then,
data to-
are